Our next speaker is Athena Lynn, and she's going to tell us about Aurora Kinases and Stentor.
Thank you so much for inviting me here to speak. I am very honored to be going after some famous professors. My name is Athena Lynn, thanks for the introduction. And I work on these amazing animals, ciliates, called Stentor. As you can see, it's going in this clockwise direction. And I guess I'm going to point. Is it? Okay. So I wanted to give you some Stentor anatomy. Oh wait, I forgot to say that I'm from the Wallace Marshall Lab at UCSF. I'd like to say that ciliates, Stentor are like other ciliates, but they're huge, and they have this feeding apparatus with the membranellar band and this apical side, sorry, it's upside down, and the membranellar band is covered in these cilia that literally suck in the food, and they can choose to eat this food clammy if they want to. And on the basal side, which is your top, they have a holdfast of which they attach to the underside of plants so that they can filter feed. Another important feature which we'll come back to later is the macronucleus. And it's this round nodulated shape, and you can see another one here and another one here as my pointer goes down. And it's in this long beads on the string type shape. And that's important for later. So I add but at the very bottom is because Stentor can regenerate. Since the mid-1900s, scientists have been performing surgery on these Stentor, cutting them in half and watching them regenerate, which in a sense this is amazing because if you cut it in half, it knows that the bottom is missing and makes a tail. And then the bottom half knows that the head is missing, so it'll make a mouth. So this is the biggest question in the lab. A lot of my lab members work on this question. How does Stentor regenerate? What are the molecular mechanisms that control this regeneration? And now we start with some very important observations. So when I sucrose shock them, they get rid of their membranellar band. And so I called this the bald state, or the no-mouth state. And the membranellar band starts forming along parallel to the axes of the body. And this extends downwards and also perpendicularly across the top. As you can see, part of it in this next stage. And then it forms an oral primordium at the basal end of the membranellar band and then moves this oral primordium up towards the top, returning to its original shape. So another important observation is that the macronucleus, shown here by these little beads, become this oblong shape at this stage. And if we make some important observations in my vegetative cells, you can see some of the same things happen. The membranellar band forms sort of medial, and it elongates, and then forms an oral primordium here, and then pinches off to make two cells. The same thing with the macronucleus that has this beaded-like structure, and then becomes an oblong shape, and then returns to its nodulated state afterwards. So once we've made these observations that regeneration and cell division have all these similarities, what better way than to do a screen with cell cycle cell division inhibitors to see what pathways overlap. And sure enough, Aurora kinase was a hit, which kind of makes sense in retrospect because Aurora kinases are really important to cell division. These are human auroras, and you can see on the top panel here that they're localized to the mitotic spindle, but they're not only localized, they actually control the formation of the mitotic spindle. TPX2 here from the paper that I stole these from helps localize Aurora kinase to their location. But the same thing with Aurora B. Aurora B actually controls the tension between the two centromeres to make sure that they're going to go the right direction. And also, Aurora B plays a role in cytokinesis, making sure that there's no centromeres in the cleavage furrow as it's coming down. And in a sense, it actually puts a stop sign up to stop the cleavage furrow from forming or stop budding it so that the chromosome can get out of there in time. So it makes sense that if Aurora kinases control cell division, then they sure might control regeneration. So thanks to the genome paper that's being in review, not being in review, it's in review, I know gratefully that I know that there are 35 Aurora kinases in Stentor. So how do we start making them regenerate? So it's much like this cartoon where we make them shed their membranellar band. And how do we do that? So I take some healthy Stentor and I sucrose shock them, which makes them shed their oral apparatus and their membranellar band to form this bald state. So I'd like to point out that only after sugar shocking do I divide these into my control and my inhibitor groups. So to describe this regeneration process, we start with, again, the no-mouth state. And this is important here because I mark this as stage 1. And then stage 2 is where the membranellar band starts forming. And remember, this extends all the way down and across the apical side of the cell. And that is still considered stage 2. As soon as I see an oral primordium form at the very bottom, I consider that stage 3. And stage 3 continues all the way until it's fully regenerated, and I consider that stage 4. Now, how do I plot the data for you to examine? Oh, I'm sorry. This all takes place underneath a microscope, so we can actually, although these Stentor are really big, we have to observe them with a lot of light microscopy. So this is the data. I've plotted for you on the y-axis the percentage of cells and then time on the x-axis. And so you can very clearly see that this stage, this no-mouth stage, stage 1, stops at 3 hours. And very rarely do I see in the 4th hour the bald state. The second state, which is called the membranellar band state, rarely goes past the 5-hour time point. And for the oral primordium state, it usually stops within the 7-hour, and then usually everyone is regenerated by 8 hours. So this is actually real data as a part of my control. Oh wait, no, this is not real data. This is my control. So let's get to some real data. So Aurora kinase A inhibitors actually shorten the time to regeneration, as you can see by this 7-hour time point. Everybody has been healed by 7 hours. So it gives some sort of uniformity to these regenerating Stentor. I can see you're not impressed. But on the other hand, Aurora kinase A and B inhibitor really completely just suppress regeneration. And of course, you can see that I'm not a very good grad student, and I completely missed the time point where the bald state was formed. But you can tell on the other side that they do eventually form in DMSO. So the next question is, how do I, the next question that I had was, is this irreversible? Is this damage permanent so they can't heal after I wash them out? So how do I do that? I take some healthy Stentor again, and I sucrose shock them, but this time I incubate them in an inhibitor for 2 hours, which suppresses their regeneration. Afterwards, I divide them into my control group. My experimental group, is to wash out the inhibitor, and the control is to leave it in an inhibitor and watch and score regeneration as usual. And you can see that on the left-hand side, that the no-mouth stage maintains for a little while, but it never passes stage 2. So it gets to the middle of regeneration and it completely stops. On the right-hand side, though, you can clearly see that they do regenerate after 10.5 hours. And so in conclusion, I'd like to say that Aurora kinases do control the regeneration time, and Aurora kinase A's speed up regeneration. Aurora kinase A and B, which is a pan inhibitor, I forgot to mention, is also suppresses regeneration, but is also reversible. I like to come back to the bigger picture, is that regeneration and cell division have a lot of things in common. I hope to parse apart those pathways and to get an even bigger picture as to morphogenesis. I hope that this has created some ideas about how the cell reinduces its development. With that, I'd like to thank my entire lab, especially Wallace Marshall. He's been an amazing PI, and my immediate mentor, Sarah, and my funding sources, the NSF and the labs and NIH. Oh, I forgot to mention that Aaron Tipton was the one who initiated the screen. Thank you for your attention.
Have you looked at the nuclear morphology in the presence of these inhibitors? Does it change what's happening with this change of the nuclear morphology?
Yes, so the nuclear morphology does change as well. So I haven't showed you the entire time course of fluorescence because it takes a really long time, and I'm still working on it.
How does it change? What does it look like?
So it does combine into this oblong shape, and then it very slowly goes back to the nodulated state.
But does is that different in the presence of the inhibitors?
Right, so I have yet to do that.
Oh.
Yeah.
So I have a question. When you cut stentors into pieces, I understand that the piece to regenerate needs a piece of a macronucleus.
Yes.
But then what's going on with the micronuclei? How many micronuclei are present there in stentor? And does the piece require a micronucleus to regenerate? Because we know that in tetrahymena, when you create an amicronucleated cell, it dies for unclear reasons.
Yes, so I think that it dies too, although in 1951, when Tarter wrote his book, he claims to say that there's no micronucleus needed, but I think it's needed.
The second question is, I wonder, when you treat, you do this sucrose shock, is this the same as, a regeneration when you actually cut off the entire structure? Because it seems to me like it may not be exactly the same situation. Because when they are shocked, then they may be resorbing the structure. So then they may still keep some less visible landmarks, like, for example, some ciliates form cysts, and they resorb all cilia, but we know that their internal cytoskeleton is still present and is still polarized. In other words, maybe it's not a true regeneration, what you see in the sucrose shock. It is actually a resorption and rebuilding the organelles, but there is no need to reorganize the positional information.
So I have done these experiments where I'm cutting them in half, and I'm getting results, but the N for now is less than 10 because I can only cut one at a time. The other thing, but we do have a machine that's being made at Stanford that will cut a number of stentor for us, and I'm looking forward to having that. The other question that you had was the resorption of the cilia. So actually, I meant to put in a video, but it was too long, that if you add sugar to the media of stentor, they actually physically shed and you can see a part of them leave, even with the naked eye.